So this is, we're going to cover basically some of the testing, not all of it, obviously, but some of the basic testing that we do for viral diseases. Viral culture is very long and tedious frequently. They're definitely longer than a very quick serology test. These can test for either antibodies or antigens or both. So just reminding you, viral immunity, just to remind you that how it works. Viruses are obligate intracellular pathogens. So they would only be able to gain access to any particular cell if there's a receptor on that cell. There's only a certain set of cells that can be infected by any one particular virus. But once they're inside, obviously they're going to take over the cell, hijack the host processes. They'll reproduce, which would be their form of metabolism, and eventually they will be released from the infected cell in a couple of different ways. It could be lysis of the infected cell or gradual. The virus actually produces a chronic infection where they produce their virions relatively slowly. And the cell and the virus sort of continue to live together. But what happens is you will get some release of some of the particles that were within the cell. They'll be released as the virions are released from the cell. There'll be a certain amount of free particles released into the host fluids. Now, we do have innate immunity, obviously, that are our first line of defense against viruses. And that includes, of course, it includes things like intact skin and stuff. When we get inside the body, it includes things like interferons and NK cells. Both of those can generally identify either infected cells, like an NK cell. Interferons is like they're released by infected cells to tell the surrounding cells to sort of stabilize their defenses. So those two things together, they're very nonspecific. It doesn't take a particular virus. They will be released and do their jobs with any virus. But then once the infected cell it releases the interferons. There are other things also that the infected cell can do, like release interferon alpha or interferon beta. These will upregulate in the nearby cells, upregulate antiviral proteins, things like ribonuclease enzymes. That'll degrade RNA, and they happen to be pretty specific for viral RNA. But they'll also increase the activity of the NK cells in the area. Now, we do have a non-cellular response to viruses, and that meaning the humoral response, and that can help the cellular response, although the cellular response really is the key thing for viral immunity. But we do find antibodies. Antibodies can participate in neutralization, preventing the viruses from binding to their receptors and causing any kind of major infection. They can also, if they bind, first of all, you have IgA at the mucosal surfaces. That will help to bind the virus directly and neutralize it so it won't be able to penetrate through the skin. Then you have IgM and IgG. Both of those can bind in the bloodstream or in the tissues, respectively. And they can neutralize as well. They can also, call in complement, right? And complement can bind, and then these things can get engulfed by macrophages and processed. But in reality, when you're talking about viruses, there's usually enough that what you really do need is that cellular response. So if the NK cell response is not sufficient, then the cytotoxic T cells are going to come in. And especially if it's already inside a host cell, antibodies can't do anything if it's inside the host cell. So the CTL will bind to the infected cell, and it'll release some not so nice things into the cell, like perforin and granzymes. So perforin will produce pores in the cell membrane, and the granzymes are proteases, and they'll be able to enter those pores. And granzymes, what they actually do is trigger apoptosis. And so that virus that's in there has just entered a cell that's targeted for very, very quick elimination. And then, so if we have this protection, why is it that we still get viral infections? Well, the reality is there are a lot of viruses, and they can rapidly divide and change their appearances. Viruses mutate very rapidly, in particular RNA viruses. And so whatever protection you had, remember your cytotoxic T cells and your humoral response are both very specific to a particular virus and to the antigens it expresses on its outside. So even minor mutations can cause those previous cytotoxic T cells to not work, and that's why you do get colds every year. Because adenovirus, it mutates very, very rapidly, okay? And then you also have some other issues going on. There are some viruses that can downregulate things like interferons. They can evade complement, or they can actually interfere with our immune response. And so, you do have like hepatitis C, that's able to block the degradation of viral RNA that interferons perform. So they can actually block the interferons. So that's the kind of stuff that individual viruses are able to do. And then once the virus is inside the cell, they can also change the way your genes are expressed. Some viruses, it depends on which ones, again, this would be individual, they suppress the later immune response, meaning the adaptive. So there's several that can do that. CMV, rubella, HIV, they all are able to downregulate MHC, which would limit or prevent an adaptive immune response. Pardon me. And then finally, there are viruses that can maintain a latent state. So what these do is they integrate themselves. Instead of just hijacking the host cell and sort of being greedy, we might say, taking over the whole thing using all of the host machinery for their own benefits, the latent virus will insert itself into the genome, and it'll just hang out there, and it'll just gradually produce the things that it needs. Or it'll just live there like a little, like a loaf, like a squatter, in your genome, and it'll use only what it needs to survive until the next time it needs it. And that latent state, that's harder for NK cells to always detect, and it probably won't produce enough antigen for either a humoral or a cytotoxic T cell response, not the right kind. You can produce as much humoral response as you want. If you've got a virus stuck inside your genome, it doesn't help you at all, okay? So this is just, we're not going to go into every single one of these, but this is just a nice chart. Nice, it's a complicated chart. You already went through all the viruses, so you can see they're here. These viruses are here. So you can see that there's an awful lot going on. And you do need to know what the causative agents could be of a particular site. Okay, so recognizing the viruses underneath these, these headings would be a good idea. Okay. There's a couple things here. You're probably familiar with pretty much everything here. The only thing I could think that you might not be, let me see, the parvovirus B19. I think we did go through that. Yeah, we did go through that that causes Fifth's disease. It's just it's called Fifth's because it's the fifth childhood disease. I think I went through that. It gives you a rash and stuff like that when you're a kid. Now, and for all the viruses that are here, obviously this ain't all of them, but these are the ones that we more commonly come across in the laboratory. This is also a somewhat older chart because one of the big ones that should be added to this, and you can pencil it in, is of course West Nile. That's a big problem in our area of the country because it's where it originally arose. In Brooklyn, New York, was the first case of West Nile that we came across, and it's a significant cause, potential cause of encephalitis. Most people don't get encephalitis when they get West Nile, but at-risk people can get encephalitis. And then, of course, we have viruses that emerge all the time. You wouldn't be, reading the news at all if you hadn't heard of Zika virus, which of course causes significant difficulties for a woman who's pregnant. And the jury's still out as to what else Zika can do. Another one, is it on here? Another one is sort of on here that is not really on here, is the Norwalk-like virus. And if you ever want to go on a cruise, you have to be aware of Norwalk-like virus. You should probably bring your own water, just suggesting. So if we go through a few of them, these are the ones that you're going to be testing sort of day in and day out, okay, in a generic clinical lab. So viral hepatitis, we have to be able to know which test. Now you've heard about hepatitis over and over again, okay? And this chart on the right-hand side here is really a good chart of all of the tests that you can see. And I want you to read it across, okay? Oh, I don't have my pointer. So if you read it across, you can see for hepatitis A, how do we diagnose it? Mostly we diagnose it clinically, but we could do serological markers. If you have IgM, anti-HAV, then you have acute hepatitis. You read it across like that. You see how I'm saying? And then if you do a total, then you're looking for immunity to hepatitis A. Remember, there is actually a relatively new vaccine against hepatitis A. I'll wait to get that. But if you eat a lot of shellfish, this would be a good idea for you, or raw fish of any kind. This would be a good idea for you. And then HAV RNA, this is actually more used in environmental testing. So they use this to test clam beds and stuff like that. Make sure it's not in there. But I really suggest that you make sure any clams that you eat are safe. Okay, here's the key thing here, the hepatitis B, you're going to be tested on this. Again, read across. Hepatitis B surface antigen, AG, watch your ABs and AGs. Surface antigen, you have an active hep B infection. E antigen, which is not always ordered, that's only ordered in certain cases. Patients who have an E antigen, there's a high degree of infectivity if they're expressing antibodies against that, if they have that currently. Anti-HBC, core, C stands core. Here you go, current or reactive acute hep B. And so you read across like this. I don't need to read the whole thing to you, so that you understand what each test does, what its purpose is. Hepatitis C, anti-HCV, current or past hep C infection. This again, hep C, they're finally using the word cured for hep C with some of their treatment modes. They wouldn't be allowed to do that if the FDA didn't say they could. And so now the blood bank has not changed their protocols for what they'll accept yet, but they may. But at this point for testing, an anti-HCV is our best test for hepatitis. And then we would back that up. We would verify that with RNA testing. Then you have hepatitis D. Remember, this would only be tested if we knew that the patient also had hepatitis B. Hepatitis D, it doesn't occur on its own. It's a co-infection. And hepatitis E, again, this one, remember this one, major risk group are your patients who are in their third trimester. This is not a common virus around these parts of the world, and it is similar to hepatitis A. Normally just gastroenteritis, except if you're in your third trimester, where then the patient deteriorates rapidly and it's pretty bad situation. You've also seen these two charts over and over and over again, but that's because they're important, okay? So make sure you know which antibodies peak during which times. Chronic, acute, when are the symptoms shown? See the symptoms here? When are they shown, okay? See if I said everything. Herpes viruses. We do a lot of testing for herpes viruses because they're not going away. Um, so we don't test for necessarily all of them, but herpes type 1, obviously this is a big one. And while we usually are testing for it as an STD, there is an issue, and you guys would be associated with that, with head and neck carcinomas and cervical and uterus carcinomas. They're related, not causative. So note this, please. Related, not necessarily causative. Okay. So, okay, you could say cervical carcinoma, somebody who has herpes virus, somebody who has herpes type 1, they have increased risk for cervical and uterine carcinomas. But if they have an STD, is the cervical and uterine carcinoma because of their association with other risk factors that would be, also related to this. So again, that's the difference between related and causative. We don't necessarily have a cause here. Some of them we know, type 8 does cause Kaposi sarcoma, etc. Okay? And then don't worry about the subgroups. That's not really important. Here's just the classic differentiation of them. I want you to know all 8 herpes viruses and what they cause. This here, herpes 1 and 2, this whole thing with oral and genital lesions, this is an old chart. There's no difference. I used to say herpes 1 was on your lip, herpes 2 was, in your genitals, but that's not true. They're kind of, they can, herpes 2 can create a lesion wherever you let it. Okay, and same thing with herpes 1. But similar lesions that they produce. These are painful lesions, as opposed to syphilis, which would produce a painless canker. Okay, but I mean to the patient, a sore is a sore is a sore. Okay, then we have chickenpox. Then we have CMV, human herpes virus 6, which gives you the roseola infantum. Same thing with 7. Epstein-Barr is 4, and Kaposi's is 8. So just to go through some of the more difficult herpes viruses, Epstein-Barr virus this causes a lot of a wide variety of clinical symptoms. Everything from what we all know as infectious mononucleosis to some pretty serious lymphoproliferative diseases and even some malignancies. Okay, so now the most common, or the most frequent, I should say, transmission of EBV is, classic contact with salivary secretions of the infected individual. Lots of salivary secretion. So while it's not wise to share a cup with somebody who has EBV, that's probably not enough saliva to transmit Epstein-Barr, which is why it gets the name the kissing disease. Okay? And that's because this selectively infects host cells that are CD21 positive. And while this component, so here's your CD21 positive cell, those cells, there are lots of them in the epithelial, a lot of these in the buccal, the glands, right, the salivary glands. Hence, you have lots and lots of virus in the saliva. Unfortunately, the receptor that's there, CD21, also happens to be a component of the C3d portion of complement. So while this will begin in the epithelial cells, it'll move from the epithelial cells, in particular in your salivary glands. It can move to B lymphocytes. And then the B lymphocytes can spread it throughout the entire RES. Okay. And then it can be the virus it can change. This can actually, you know, cause significant changes in the immune system. The B cells actually become potentially polyclonally activated, meaning they'll start producing a number of antibodies, not just EBV antibodies, but you'll get your heterophile antibody, you'll get autoantibodies, those are your cold agglutinins, you'll get rheumatoid factor anti-nuclear antibody, you get all kinds of crazy things that can be associated with these polyclonal B cells, okay? And the virus can kind of hang out in those B cells, giving you this latent infection, and every once in a while they'll activate and start just throwing out all those viral, they'll start producing viral particles. The process of producing viral particles always sheds many antigens because viruses are fairly sloppy in their construction techniques, okay? And then it's in the variety of antigens, right? The types of antigens that are secreted, that are thrown out, where we can associate, which we can associate to different clinical stages of the viral infection. Okay, and that's where this chart comes in. Okay, it does change. You get different antigens thrown out at different phases of the disease. Okay, obviously uninfected, you shouldn't have any of them. Good luck with that. But then when you start going through each of, so IM means infectious mononucleosis, we're just going to give you the classic findings of EBV, right? So classic infectious mono, here you go. You've got a positive IgM, you've got a positive heterophile antibody. That's very helpful. And you've just diagnosed infectious mononucleosis, classic, okay? But eventually people get over infectious mono, right? They do get better. So when they're convalescent or past infection, what does it look like? Okay, no more IgM or heterophile, but you still got the IgG and an anti-Epstein-Barr nuclear antigen. Get it? Right there, see? Okay. And just a note, the VCA is a viral capsid antigen, and EA is the early antigen, okay? Now, these are people that get better, but we also know that some patients with infectious mononucleosis, they don't really seem to get completely better. They kind of produce this chronic infection that sort of recurs. So they get this, oops, sorry, break in my office. They get this post-infection, I'm sorry, chronic infection that sort of just keeps giving them recurrent symptoms. And you can see the levels of the antibodies are much higher, especially for the viral capsid. And then the early antibodies persist. And your nuclear antigen, that antibody, that doesn't persist. So, or at least there's not enough research to verify that. So you get this strange chronic thing going on. Okay, and then you have several other issues going on, lymphoproliferative diseases, Burkitt's lymphoma, and nasopharyngeal carcinoma. Now, these obviously are not used to diagnose any one of these three issues. However, what they do help to do is support an EBV role in these issues, okay? So that's important. Other things that you would find in the laboratory, this is just the serology that you would find. But other things that you would find for any of these, infectious mono, any of those, your lymphocytosis increases. You get an absolute lymphocytosis of about 50%. So that's high. We don't usually expect it to be that high. With 10% atypical lymphocytes. That's strange. Okay, we don't usually see atypical, but viral infected cells, that makes sense. And then, of course, looking at your heterophile antibodies and your EA, all of this together is needed to make a definitive diagnosis of infectious mononucleosis Epstein-Barr infection. Now, clinically, what is your patient going to be experiencing? Well, clinically, they're going to have, I don't know if, pardon me, any of you have ever had infectious mono, but basically you're kind of run down for a long while. You have a fever, your glands are swollen, you have a sore throat, and you might have an enlarged spleen, or liver even. That's not a good thing. And even your eyes, that's one of the things that they see is you have this swelling around your eyes. It makes you look kind of run down, okay? Now, they're not unique to infectious mono, but they do correlate well. And that's the problem with infectious mono. It's kind of, it's just that you're sick for a long time, but like a month. Usually you're pretty wiped out for 3 or 4 weeks. But other than that, there's nothing it's not viral specific. Okay, then we talk about another herpes virus, cytomegalovirus. Now this, you already know, this is pretty widespread, right? Most of us adults have already been exposed to this, okay? And unlike EBV, where we know that this is at high levels in saliva and not as high in other body fluids, CMV, you can find pretty high levels in anywhere: saliva, urine, stool, vaginal, cervical secretions, semen, breast milk, blood, pretty much everywhere. Okay, so this is a, you know, this is a disseminated virus. And most of you can't think back into your history and go, oh, I remember I had CMV when I was 6. It doesn't work that way. Most of the initial infections with this start out as asymptomatic infections and frequently self-limiting. So you might not ever realize that you had this, okay? This will not produce a heterophile antibody like EBV would. So the heterophile antibody is specific towards EBV, okay? But the illness will look similar if you do develop symptoms, will look similar to infectious mono. You'll get the fever and the fatigue and all that stuff, okay? But that's really a relatively small number of cases. Most cases are pretty, you know, low-key mild. You might not even notice it. You might think you have a cold or maybe an allergy for a day or two, okay? But unfortunately, where EBV sometimes produces a latent infection, CMV does produce a latent infection, and that latent infection will be set up within the monocytes, as well as like dendritic cells and any of the myeloid progenitor cells. And what happens then is that that infection can become reactivated later in the patient's life, okay? And for certain patients, in particular immunocompromised patients, your HIV patients. Now, obviously with a neonate, it wouldn't be a reactivation, but for these 3 patient populations, CMV reactivation is a serious issue. The other patient population is the transplant patient, and that's because you're going to turn them into an immunocompromised patient if they weren't already when they started this process, okay? And so finding CMV negative patients becomes really, really important in the blood bank, okay? Now, if it reactivates, what does it look like? Well, then it's a little bit more serious because of the patient population. Patient's going to have a fever. Their white cell number is going to drop because all these viruses are, popping out. They could end up with enlarged liver, pneumonitis, GI complaints, and retinitis. These are all pretty common findings. And also, one of the big things, of course, from the lab point of view is allograft failure. So whatever you try in a transplant, it's going to fail. Okay. Now with the neonate, we already know that this is a special case because, of course, it's not really a reactivation in the neonate, but they don't have an immune system to deal with this infection if it's vertically transmitted to them. So they're not being, it's that they got it from their mom. And usually what happens is the mom acquires the infection while pregnant, and then it's vertically transmitted. If I already have it and then become pregnant, right? I have a chronic case and become pregnant, that's not going to necessarily transmit unless the virus reactivates during my pregnancy. So about 10% of infants who are born with congenital CMV will be symptomatic at birth. So even many of them will be, asymptomatic. But the symptoms, when they occur in an infant, they are usually related to platelet dysfunction and central nervous system involvement. So they're obviously much more severe. You get things like jaundice, hepatosplenomegaly, petechia. You get all kinds of neurological abnormalities that can occur, anything from just plain lethargy all the way to microcephaly. And in that 10%, there's a mortality rate of 5%. So that's not insignificant. And then 50% of those infants that survived, okay, they're going to develop sequelae that persist. So what does that mean? So the sequelae is kind of like scars. So you're talking about hearing loss, visual impairment, mental delay, those kind of things, usually CNS issues. Now, that was the 10% who were symptomatic, right? Let's talk about the 90% who were asymptomatic at birth. If they're asymptomatic, how do you know if they have it? You don't, okay? But unfortunately, those 90% who are asymptomatic, there's a percentage of them who, even though they were asymptomatic, so clinically you couldn't see anything wrong with them, they will develop issues later as they mature. 10% of these asymptomatic patients will develop hearing loss. That's basically, it's slowly progressive. Between the first 5 and 10 years of their life, they'll actually go deaf. So they won't start out deaf, but they will go deaf. And then that's 10% will develop that. A smaller percentage, not quite completely fixed at a number yet, can develop other neuromuscular or central nervous system defects. Not sure, that there's a wide range of them. Okay. So those obviously are major issues. So how do we detect it? Now, is CMV tested for at birth for every neonate? Not necessarily. And one of the reasons is we can't do anything. We don't have a cure for cytomegalovirus. It doesn't exist. But if we want to know if the patient is born in distress, or even if you have a patient, immunocompromised patient HIV, how do we detect it? Viral cultures, one, we do CMV cultures not infrequently. We do antigen assays not infrequently. And then, of course, really what we're moving towards are those molecular assays and serology that help us to really identify these infections. The preferred method at this point is viral culture. So that you can demonstrate viral antigens or DNA in the specimen. That's the preferred method. So you can get any kind of specimen, urine, blood, whatever it is. Very easy to collect urine on a neonate, so you could probably get urine specimen, and they'll do a culture off of that. The traditional culture that we have for CMV, we have to look for those CPE, those cytopathic effects, on a fibroblast cell line. That can take up to several weeks. And that's the traditional method. The reference method for detecting congenital CMV, what you do there is you isolate the virus, and with a culture, you do a viral culture within 3 weeks of birth from either the urine or the saliva, and you're going to be looking for those cytopathic effects. But that can take several weeks, 3, 4, 5 weeks. So what they've done is they've enhanced it with a shell vial, where shell vial just basically adds centrifugation to sort of force the virus onto the cells, speed up that process. But what it does is it really reduces your recovery time. Instead of weeks, because CMV is a lazy virus, maybe I'll infect you, maybe I won't. That's the reality. But if you force it, say get in where you go, it can take, you reduce the recovery time 16 to 72 hours. Okay. Now, uh, that's just makes you aware your cell line will then have infected cells in it. You're not necessarily producing live virion, which is something that usually the traditional culture requires. In this case, with a shell vial, once you have an infected virion, we can use a tagged monoclonal antibody to detect those early antigens that are being produced within those cells. And that's what's done in the shell vial testing. There are proteins unique to CMV. There's a PP65 protein, which is a lower matrix protein that shows up in the peripheral blood. That is also another way, molecular test for speeding up detection of CMV. And that's done with either immunochemical or immunofluorescent staining. And then, of course, we do have PCR. PCR is most commonly used with more sterile samples, things like CSF or amniotic fluid if it was collected. You don't usually do a PCR on a specimen like on a blood that there's just so much stuff that they would have to weed through to get to it. The serology that we do for CMV, most of the serology is limited to donors for blood. So we look for IgG, which tells us that there's a past infection. And we can automate these with an EIA that's on a either on a microtiter plate or a microparticle system, so you can automate it and you don't have to do it a single test at a time. But frankly, we don't look for CMV in the average patient. We only look for it as a general rule in blood bank donors to see if we're spreading it. And remember, we don't test every unit. We look for those negative units, and the purchaser has to pay for the extra testing. But even though we don't test many, you can see the consequences are pretty widespread. There's a lot going on here with the CMV infection. So it's one of those things that you say, well, maybe we should test a little bit more. Varicella zoster, or chickenpox, otherwise known as chickenpox. This virus has changed drastically in the last 20 years, okay? Not so much the virus itself, but its role in our community. Let's say it that way, right? The epidemiology of it. So when I was a kid, everybody got chickenpox. And if you didn't already get it, by the time you were 5, you would go to school and you would get it at school. And so kids would be out, for a couple of days with chickenpox. It happened all the time, okay? If you get it as a child, it's pretty mild, self-limiting infection. I mean, relatively mild. Having the pox can be, somewhat traumatic, and the kid, as long as the kid doesn't scratch or poke at the pox, they should be fine. And they'll be, there's only 1%, so we're going to say that this is a fairly mild disease. However, it is not a single time that you get it, right? So chickenpox will set up a chronic infection inside your peripheral nerve cells, and that virus can reactivate and cause a secondary infection later on in life, usually later on in life, and that infection is called shingles. Now, generally speaking, shingles is more serious. It still just produces a pox, but the reality is this pox from shingles is much more painful than the pox from chickenpox. Chickenpox is itchy at the worst. Shingles pox are painful, very painful, okay? And while certainly you don't want shingles, what we have done now is we have developed a vaccine in 1995 that's given to all children, which reduces the number of primary infections that you have. So the patient is given a mild form of chickenpox. They can potentially get chickenpox, but even if they do, it will be a very mild case. Pardon me. One of the characteristic things about the pox from either chickenpox or shingles is that they almost always start on the trunk of the body. Classic case for chickenpox is on the belly. Classic case for shingles is a little bit on your flanks, right? So that's classic. And it kind of starts there and reaches all the way up to the rest of the body. Now, if you get this the first time you get chickenpox is when you're a pregnant woman, it can affect the progress of your pregnancy. It can lead to a premature birth. It can also lead to congenital malformations if the infection occurs in the first trimester. So that's something that we would have to worry about. And we would also have to worry about immunocompromised patients, since they're much more likely to develop a disseminated disease where the rash is much, much more extensive and neurological implications are much more likely. They can even develop things like pneumonia or hepatitis. Okay. Now, you wouldn't know it from the number of shingles vaccine ads that are out there, but between 15 and 30% of people will experience a reactivation of the virus. Most likely the reason that you develop this is because you lose that the memory, right? The immune memory as you age. It's possible. Okay, and so you do get that shingles rash. The other problem is, while chickenpox is self-limiting and the rash usually only lasts like a week to 10 days, shingles on some people can last months, and it can be incredibly severe, okay, and incredibly painful. And it's, of course, much more problematic in patients who are immunocompromised. It is possible that you can develop complications which can become quite severe. One is called herpes ophthalmicus, which can lead to blindness. It's basically an infection in your eye. Now, that's not life-threatening, but it's obviously blindness is not good. Pneumonia, which is life-threatening, and visceral involvement, much more common in immunocompromised patients. So in those patients, that kind of complication would be life-threatening. Same with the elderly. So, okay, so how do we diagnose it? Well, there are several things that we can do. One of the things that's noticed, if you look in the base of the vesicle, this is the histology that might be done by, I have never seen a pox removed and sent for histology, because they usually just say, hey, they point out and say you have chickenpox, don't scratch. But it is possible, if you did a histology from the base of the vesicle, you would see this Zank cell, okay, which is a giant multinucleated cell. The stain here is a hematoxylin eosin, or you could see it in a right Giemsa, you could see it in a toluidine blue, or a Pap stain, okay? Now, this is any herpes virus. So the pox that you get this from, that could be a herpes pox, it could be a varicella pox, both are herpes, right? But it could be a herpes simplex, or it could be varicella. So it's helpful. It tells you that it's herpes and it's not syphilis, but it's not specific, okay? So instead, we have other tests that help us to be more specific. We have, obviously, direct immunofluorescence staining. We can do monoclonal antibodies directed against varicella zoster, and we can stain our histology slide with that. You'll get very specific, very rapid results. We can do a viral culture. Now, viral cultures are always going to be more time consuming, and if the patient has a low viral load for this particular one, we don't necessarily get great results with viral culture, but it's there. You could do it. Obviously, you can do PCR, obviously most accurate, most sensitive method, and it's very useful if you have some kind of atypical presentation. Like maybe you do have nervous system, central nervous system symptoms. In that case, maybe PCR is the best for you, because the other techniques are not necessarily specific enough. Now, the serology It's not really all that helpful if you need to detect a current infection, so meaning with shingles. But we can always look for a fourfold rise in antibody, but that would require that the patient come in obviously acute and convalescent. Basically, what we use the serology for is to demonstrate what we would call, um, some labs did used to call it immunity, but it's not really called immunity. Basically, we're looking for protection for especially for our healthcare workers. If you come from a country where chickenpox is not endemic, so here in the United States, it's not now, but it was endemic. So all kids got it. It was pretty much normal. But if you come from a different country, Philippines, for example, chickenpox doesn't occur there. So you move here, you decide you're going to work in the hospital, you go in, you start working in the ER. A patient with chickenpox comes in, they're 5. It's pretty self-limiting in them. If you get it at 25, 35, 45, it's quite serious. Even at 15, it can be quite serious, very, very debilitating disease. It'll look a lot more like shingles. And so we want to protect our healthcare workers from that and know which of our healthcare workers shouldn't work with these patients who come in with these pox and which ones can. Or actually at this point, we would just give you the vaccine, right? So if you have never had chickenpox and you test serology negative, we would give you the vaccine, okay? Most of our screening tests in serology will test for total IgG, but remember, one of the things that's important to remember with varicella, if you're using a serology test that's like a card test, which is a pretty common test for us to use, you have to really watch out for the prozone effect. Varicella is very, very antigenic, and so you can get very, very strong reactions where it completely neutralizes your reagent. So make sure that you always run this. If you run it at a dilution, which you always should, and it's negative, increase the dilution factor. So if you run it at a 1 to 2 and it's negative, run it at a 1 to 4 to be sure. Okay, that way you know that it's been repeated using a higher dilution, and you know that it's definitely negative. Okay, poliovirus. This is, remember, poliovirus has been uh, very significantly reduced in our country, right? We have had a very good campaign against this particular virus. But one of the things I want you to note is that poliovirus isn't as scary as the history would tell you. About 90 to 95% of infections asymptomatic. You won't see anything. Unfortunately, in that smaller percentage, you can have symptoms, right? The majority of that 5 to 10% that have symptoms, it's going to be relatively minor. They'll look like they kind of have, a rough cold, right? Something, maybe the flu, fever, headache. They'll get sick to their stomach. It is mostly a GI infection, diarrhea. They might experience neck stiffness and pain in their arms and legs, and that's pretty common. Okay, but they'll heal. Okay, and they'll heal in a couple of weeks. It'll be a little bit prolonged, but in a couple of weeks they'll be completely better. It's only about 0.5% of all the cases of infection where you end up with any kind of muscle weakness or inability to move. Now, this can occur progressively, or it can be rapid onset. It most often involves the legs, but it can affect other areas of the body, including the head, neck, and the diaphragm. Okay? Even of this 0.5%, many will recover. Okay? Even fully recover. It is only a very small percentage where you see patients who develop what we call infantile paralysis, so their leg or their arm becomes weak and flaccid, and it doesn't continue to grow. You have even less of a percentage where the patient's diaphragm is affected and they are unable to breathe on their own, so they have to be on a respirator for the rest of their life. So this is a virus that made the news because of the size of the outbreak in the United States that occurred, that led to, it wasn't a larger percentage, but because the overall outbreak was larger, it seemed like it was an awful lot of people. Okay. So this particular virus, this is an RNA, single-stranded RNA virus. It's an enterovirus, so it's a fecal-oral kind of situation. Pardon me. Normal infection occurs in the gastrointestinal tract, and only in that small percentage do you get any spread of the virus to the central nervous system. It would have to go to the central nervous system in order for you to have any of those symptoms, neck pain, muscle weakness, anything like that. And this is an RNA virus. It'll colonize the gastrointestinal tract temporarily, and then eventually it'll be over. Only in a small percent, certain serotypes will be able to move to the central nervous system. So this outbreak that occurred historically was significant enough for many researchers to work on it. And so you have two vaccines that came out of the frenzy that was around the poliovirus outbreak in the 30s and the 40s. And we had, or 40s and 50s, I should say. We had the Sabin vaccine, which was an inactivated vaccine. So Sabin took the virus and basically fixed it with formalin. It's an inactivated, it can't infect any cells. Pretty simple process, and it worked. But that's an intramuscular injection, and you don't get the virus in your muscle. You get it in your gastrointestinal tract, so you get slightly weaker coverage, right? Protection from the intramuscular or the Sabin vaccine than you do from the Salk, S-A-L-K vaccine, which is an attenuated virus. Salk just passed the virus through, I think it was monkeys, over and over again. And he came up with a couple. The second one that he came up with, he decided, oh, this is the one that I'm going to use. And with this, it's an attenuated vaccine, causes a weak infection. But remember, this is an RNA virus. So what do RNA viruses do? They mutate. And if I give you this vaccine, the Sabin vaccine, which is an oral, it's a little drink that you have, but you have to be over a year old in order to have this, the oral vaccine. When you drink that, it produces a live vaccine, a live infection in your GI tract, and you end up potentially the virus can mutate back to its wild type and cause polio. In the United States today, the only source of polio infection is actually from the Sabin vaccine. And that's because of the tendency for RNA viruses to mutate. If it mutates back quickly enough before you develop any immunity, that's where you can develop polio. Okay, and you might say, how often? I've seen one case in my career, which is, I think, kind of a lot considering there's very few cases of polio at all in the United States. Okay. Now, lab diagnosis is very rarely needed because it's pretty, the muscle weakness and the flaccid nature of the muscles is pretty characteristic. One of the biggest problems is that doctors don't recognize it because they haven't seen it. Routine labs, right? Local labs don't do this, generally speaking. Viral culture is what you would need to do. You grow it up in a culture and then you do PCR. You can do PCR on, pardon me, I'm sorry, the culture. What specimen should you use? You should use a stool specimen because this is a GI infection. If you do have evidence of neurological effects, then it is possible you could send a CSF for viral culture as well. Okay, and then PCR can also be done from either of those specimens. So one of the things that we frequently do have to run in the laboratory are some serologies to help screen for the presence of the routine viral infections that little kids get. Okay, whether it's for the little kids, see if they have those infections, or if it's to maintain hospital safety. It could be literally, again, these are the viral infections of childhood here in the United States that we're most commonly used to. So if people come from other areas of the country, we want to know what their risk is in this new environment for them. And so it helps us to determine immune status of our patients and of our employees, people who are going to work, and it can also determine whether or not they're eligible to work in certain areas, because you don't want them to get sick. So that's one of the things that we generally do. What we are really trying to look for normally with these tests is to see if the patient has immunity. Now that immunity could come from having had the infection, or more likely in the United States, having had the vaccine. So we're really trying to see, okay, is the patient going to display characteristics of immunity, or are they at risk? And so for some of the tests, our actual result will be immune or non-immune. Varicella, you might think, varicella is different because that produces a chronic infection. So you're technically never immune to it because you can always get it again. The difference is that you actually get it from yourself, right? So it's your own infection reactivated. But for something like rubella, you can report it as immune or non-immune because the patient develops an immune response. That's basically what you're saying. So the ones that we look for are rubella, rubeola, mumps, varicella. Again, that's the one we don't report as immune or not immune. These are the 4 big ones that we look for. They're pretty much covered on every, employee health exam, stuff like that. And we're trying to determine if you're protected, if you're going to be safe where you're going to work, and also if you're going to bring anything to the table, because if you're not immune to it, you could get this disease and bring it into the hospital. So those are all things that have to be considered when any employee is considered for working in a hospital. So when we look at this, I just added this chart here just to demonstrate the vaccine coverage this is just from C94 to 2013, and you can see, pardon me, that there's been pretty consistent upswing. There was this downswing here for the Haemophilus influenza B vaccine. Here, the purple one, you see that here. That one has seen a downswing, but I think that might have had more to do with manufacturing. I'm not really sure what exactly caused that. Even with all of the don't get your vaccine stuff, there still is a general uptick in these. The only one you can see that has flatlined a little bit is the MMR. Okay, and we don't have 100% compliance on that. And a lot of that has to do with people not realizing how really how dangerous these bugs are. Okay, so then we have rubella. So if we go through each one individually, this is, remember, an enveloped single-stranded RNA virus. It's from the family Togavirus, genus rubivirus. Isn't that exciting? This is transmitted via respiratory droplet or vertically during pregnancy. And you probably know it a little bit more commonly as German measles. For most of us, it's probably a benign, self-limiting disease. You're uncomfortable, you're not feeling well, but it's for an adult, it's not really going to be a terrible thing. However, in adults, it'll just similar to chickenpox. In adults, it is a more serious disease. It's more painful, it's a bigger rash, the whole thing. Not in all of them, but if you get it, you'll get this rash. And the difference with rubella or German measles is instead of the trunk being first, it starts on the face and then it moves to the trunk. But that rash will be, it'll take about 3 to 5 days, it'll go away. You'll have a fever, a little, swollen glands, the whole thing that usually goes up, seems like an upper respiratory infection. Rarely it can cause encephalitis, and the encephalitis it would cause would include thrombocytopenia, potentially hemorrhage, neuritis, those kind of things. So that's a rare complication. But if you get rubella during a pregnancy, the consequences are severe. Everything from rubella syndrome, miscarriage, stillbirth, all of these things, terrible, especially if the infection occurs during the first trimester. Okay, and the characteristic signs of rubella syndrome or congenital rubella syndrome (CRS) include deafness. There are eye defects, as you can see in the picture there. You can see the cataracts. Also glaucoma. I mean, to see that in an infant is pretty abnormal. And then there's cardiac abnormalities, the PDA or patent ductus arteriosus, where you have the connection between the pulmonary artery and the aorta. That's not supposed to be there, okay? And on top of that, there's microcephaly, which would include things like mental delay, motor disabilities. Any combination of those, that's also triggered with a positive rubella test, and you know that the patient has congenital rubella syndrome. In order to reduce the frequency of CRS, vaccine was developed. It is a live attenuated vaccine, and that was developed really because of the risk to the offspring. And it has become part of the triple vaccine that most kids get called the MMR, measles, mumps, rubella. Okay, so most kids get it when they're young, and then they want to go work at a hospital. We do a rubella just to make sure that you actually did develop immunity to it. We don't test other patients routinely. Okay, but we can also identify active cases, because as you get an increase in the number of people who don't want to participate in vaccines, that doesn't mean that the virus goes away. Just because 9 out of 10 of your, one of the things that people sometimes don't remember is the vaccine develops immunity in me, and that will protect me from a severe infection. But that doesn't mean that the virus can't get in me and I can't travel. Like, I can't still be a slight, carrier of it. I'm certainly not going to spread it like I would if I had an infection, but it doesn't mean I couldn't participate in its spread tangentially. So 9 out of 10 kids in your kindergarten could be protected. That's great. That doesn't mean that your kid is protected by their protection. It doesn't work that way. Now, the live vaccine does have some benefits. If you get it when literally you get it, you actually spread some of that immunity during the time that you have the vaccine, but not 5 years later when you're going to school. That doesn't happen. Okay, so we test for the IgM and the IgG. Serology, and when we're doing that, we're testing, is this patient protected? If we're looking at IgG, we're looking for immune status. If we're looking at IgM, we're looking for current infection. If we do need to verify that the patient might have something, or if we suspect rubella syndrome in a patient, we can do a viral culture. Pardon me. Certainly, that's probably the best way to do it. While anyway, you don't need to know all that stuff about CPE, I don't think. But it is very labor intensive, and it'll take time. So there are other mechanisms in order to test it. Serology is going to be your best bet for doing for identifying a common a current infection. Pardon me, a current infection. Although there is a PCR test available. to verify IgM levels. Rubiola, another one. This one is spread also by aerosol and droplet. It's a single-stranded RNA virus from Paramyxoviridae, and it's very infectious. This is one of the reasons that this one was targeted for vaccine development. This is the most infectious of all the childhood diseases. If an unvaccinated person comes in contact with an infected person, there is a greater than 90% chance of getting the disease. Okay, so it might take a little time. This has a 10 to 12 day incubation period. And it might not seem, too crazy. The patient will develop a fever, cough, runny nose, and they'll get conjunctivitis. And it'll last for like 2 to 4 days. That's the period when you're infectious. Okay, so keep your little eye infections at home. And one of the ways to identify it are with the Koplik spots. And these spots are these tiny little white spots on the inner cheeks or in the lips. They're grayish to white lesions against a bright red background. So you have two things happening here. The mucosal membranes are filling with blood, right? They're bright red, but you get these little pustules, these little spots that appear on that bright red background. And they'll stay there for several days. This is not unique to rubeola, but it is present in rubeola. Okay. The patient will also have a rash. This one also begins on the face or the head, and it'll spread to the trunk. So you see varicella starting at the trunk is a little bit more characteristic. And that rash will last about a week or so. It is not uncommon to develop complications, secondary infections, things like that in any age person that might get this, but specifically in the immunocompromised, of course. They can develop croup, bronchitis, diarrhea. There's all kinds of things that can be secondary to this. Okay, by far it's a rare one, but it's the scarier one is subacute sclerosing panencephalitis, or SSPE. It's a rare complication, and what happens is the virus is persistently replicating in the brain, and it's very rare, but it is one of the complications that occurs from rubeola. If you develop this one, this is measles during pregnancy. Premature labor is probably one of the scariest ones. Spontaneous abortion, low birth weight. These are all pretty scary things that occur with this. But the reality is, one of the things that you need to know about rubeola measles is that this is a high mortality rate for people around the world, okay? Especially in children. So this is the scary one. This was still number 3 killer in the world of, not too many years ago. I haven't checked lately. So this is a very significant problem, and it's in particular in areas where they don't have the vaccine. Okay. Mumps. Mumps is also transmitted respiratory droplets. Potentially this one might be through fomites because it is pretty high concentration in saliva. So like when you put kids, and they have those ball pens and stuff, it is potential that you could spread it that way. The incubation of this one is 2 weeks to 18 days. And it'll spread from the blood to the various tissues, including the brain or the meninges, the salivary glands, testes, ovaries, and it'll produce inflammation. The inflammation of the parotid gland is where, the salivary gland here, is where it gets its name. And this is actually a pretty generous picture. These can get very large, really swollen. You can barely see them swollen on this poor little girl, or the cartoon of the little girl. But the reality is they can get quite enlarged, and in old pictures, you might have seen this little girl would have like a little cloth tied under her chin up over the top of her head. And the reason was her parotid glands got so enlarged and heavy that they're pulling on all her muscles. And that actually causes quite a bit of the pain. So just tying them up, it's not that you need to put ice or anything like that, tying them up so that you have something else other than just the muscles in your jawbone holding them together, it did relieve some of the pain. Here we go. But that inflammation occurs in about a third, 30 to 40% of the cases, and they end up with, it'll resolve, but it'll take about a week to 10 days to resolve. So that's a long time to be in pain. Other complications of mumps include things, because remember, it can move to the meninges. So meningitis is one of them. About half of the cases will develop asymptomatic meningitis. So you'll have this swelling of the meninges and not know it. It can still leave lasting effects, which is why that's still not good. Symptomatic cases, 10 to 30% will have that. Testicular inflammation occurs in anywhere between 20 and 50% of males, post-puberty aged males. And this is significant because this inflammation can lead to infertility. Ovarian inflammation in only about 5% of post-puberty aged females. So that's a relatively low percentage. Deafness, however, 1 in 20,000 cases. So that's that's not insignificant considering the number of people that how infectious this particular organism is, or virus is. Okay, before we had the vaccine, this was the most common cause of deafness in kids. I don't know, if you look back in your family line, you might find one or two. I can think of already one right now who's still alive that this is how he lost his hearing. He lost only one ear, but he lost it. There is, if you develop mumps during pregnancy, there is an increased risk for fetal death. Again, you can just imagine all that swelling, how bad that would be. But there are no congenital syndromes associated with it, okay? And as far as our diagnosis, certainly we can do a viral culture. That certainly can happen. And you send us, a specimen. Saliva would obviously be best, but we could do it from urine, CSF. We've even done it from, they've even done it from a swab of a parotid gland. That's easy enough to do. We do have a shell vial technique that uses rhesus monkey kidney cells or a human embryonic lung fibroblasts. And then they can do monoclonal antibody labeled with fluorescent label to detect it more rapidly. Even more accurately, we have reverse transcriptase PCR done on specimens from the buccal cavity, the throat, CSF, or urine. But the simplest, the most practical one really is the serology. If we can demonstrate elevated IgM levels or see an actual 4-fold increase in IgM levels, in the IgG levels, pardon me, then we can say that we have mumps. We can diagnose it as that. There is some cross-reactivity between mumps and other paramyxoviruses, but if you use your serology plus your clinical signs, you'll be fine. Okay. Rotavirus, fun virus. Rotavirus is an enteric virus. It infects the GI system. It causes a significant or severe, well, it depends on who it's in. It causes pretty severe gastrointestinal illness, gastritis, with diarrhea, vomiting. The worst cases are in the immunocompromised, the very young, the very old, okay? This is a significant problem for preemies because You lose so much water, you lose so much of your body weight that this can be fatal for preemies who don't have weight to lose. Um, but and it's also highly, highly contagious, okay? It is a fecal oral transmission, but it also has been associated with fomites and it has an extended survival on surfaces. So it's very important to make sure that everything is washed, okay? But if you're dealing with a kid who's puking and crapping everywhere, it's very difficult to keep yourself as clean as you normally do, and that's how it usually goes through families. Now, they have a vaccine now. It was introduced in, when was it introduced? I don't remember, not too long ago. Before the vaccine was developed, rotavirus infection was responsible for 200,000 emergency room visits, 55,000 hospitalizations, and 60 to 65 deaths a year in the US. Okay, worldwide, it was the leading cause of severe diarrhea in young children. 2 million hospitalizations worldwide, more than 500,000 deaths of children ages 5 and under annually. That's worldwide, okay? Now, older kids and adults, we can get it. We don't have the same risks because we're heavier, first of all, and we have a little bit more ability to get through these things. But you lose so much water so fast that it is very difficult for young children and especially the very small to survive it. It's also very highly contagious, okay, which I think I already said. Pardon me. For us in the lab, here's the vaccine. The vaccine became available in 2006, and by 2009, it was added to the immunization schedule. So what's our lab testing? Well, they have several little kits available, and you can see that these are the best. The child will produce poop. If they're not producing poop, they don't have rotavirus. They will produce an enormous amount of poop. They're just losing water so fast. And we have several different things. We have these like little immunocards, these lateral flow. They're both lateral flow technique. This is in a little cartridge case, and this one you use the strip in there. They both take about 10-15 minutes to perform, and so you have very quick results, very simply to the floor. Okay. In some cases, they add this. They add like a couple of viruses together in a kit. And some hospitals like that because they can screen for, say, rotavirus, adenovirus, those kind of things. Norovirus, they can screen for all of them at once. This is an easy one. You see this a lot in areas with pediatric ERs. Influenza. We know a lot about influenza probably already. It's a segmented RNA virus, which is what makes it so unique. And it is of the family Orthomyxoviridae. Okay. There are 3 strains of influenza that circulate in our area commonly, and those are influenza A, B, and C. But there's nothing to keep any strain from circulating here. And you will find that from year to year, there will be an occasional strain that is so novel and so new that it causes a lot of damage, which is the case this year in 17-18. It's a pretty significant year. What happens is the scientists and the epidemiologists attempt to predict which flu strains will be circulating in this particular area of the country, and they produce a vaccine that will cover for it. Unfortunately, they don't always get it right, again, as of this year. So these are the kinds of things that happen, and we end up with being hit by the flu. You get lots and lots of specimens in. What do I have? I have the flu. Go home. There's nothing we can do for you. Okay. Most of the tests that we perform in the laboratory look for influenza A or B, and that's because those are the 2 that cause the majority of the most significant disease. C can circulate in our area, but it rarely does circulate in our area. So since you do have to limit what you can test for, you limit C, you remove that out. So the common thing that's done now is called RIDT, or Rapid Influenza Diagnostic Test. It's basically an enzyme immunoassay. It's a lateral flow. And there are automated systems as well, which are immunochromographic assays for influenza nucleoproteins. So you're specifically looking, there's only a few proteins that the virus is going to make, and it'll be shed into the surrounding area in the upper nasal cavity where the infection is. So you take a specialized brush that has a wire shaft and you place it all the way up and around into the back nasal cavity of the patient. This goes in viral transport media, you send it down to the lab. We have little strips like this, we have cartridges, and then we have magic box machines where you put the specimen in the cartridge, dump it in that thing, and the computer reads the whole thing. It just spits out a result. There's all different kinds of things that go on there. All of these different proteins are from recombinations of the variety around these two, neuraminidase and hyaluronidase. And the constant genetic variety that's developed in these leads to a lot of the changes and the lack of total immunity that we have. You can have reduced resistance, but you don't have immunity to the flu. RSV, this is also RNA virus, single-stranded negative sense RNA virus from Paramyxoviridae. It comes from the fact that the F proteins on the surface of the virus cause the cell membranes of nearby cells to merge, forming what's called a syncytia, or a multinucleated cell. So multinucleated cells would be something that you might find in the secretions of a baby that has RSV. Okay, the virus infects the lining of the respiratory tract, the lungs, the breathing passages, and adults usually they'll think they have a cold. It's really not that big of a deal. However, for infants, adults, and the immunocompromised, RSV can be devastating. Okay, it is in kids, RSV is the most common cause of bronchiolitis and pneumonia in kids under the age of 1 in the US. It's also, they're recognizing it as a significant cause of respiratory illness in adults, all of us, but we soldier on, as it were, okay? The key group here that we're worried about, again, premature infants. These ones, especially if they have some kind of airway compromise, maybe they have asthma, maybe they have some kind of, they're born premature, they have an airway problem. So because of that, they have a greater chance of having a more severe infection, like a lower respiratory tract infection, okay? And so this is a lower respiratory tract infection in a preemie is a significant problem, especially viral. We can't treat it with anything, okay? We don't have antibiotics. I mean, you can use antivirals, but the kid doesn't have much of an immune system to work with. So for the most part, what we use are antigen detection tests. IFA, DFA are old-fashioned. We don't use those as much anymore. So these were slide tests where we literally made a slide of the bronch wash and use a fluorescent labeled antibody. Those are old-fashioned. So now we have rapid antigen tests, little cartridges, slides, magic box stuff, and we also have culture and PCR. This is the stuff, one of the biggest reasons to figure out if your patient has this, so that you can prevent an epidemic in your nursery. You definitely don't want this in your nursery. So this is important, okay? These antigen detection tests the sensitivity is pretty high, 80 to 90% on these, and they're pretty reliable in young children. Not as reliable in older kids and in adults, but with little kids, this is what you'll test for in your laboratories. Human papillomavirus, HPV, this is a DNA virus, a little bit different, and it's capable of infecting any human being. And like all papillomaviruses, HPVs produce infections in keratinocytes, right? That's their host cell. So you'd find those on the skin or the mucous membranes. And most HPV infections will actually be subclinical. You won't see them at all, cause no physical symptoms. However, some people will develop warts, okay, or papillomas, which is a wart. And those warts, potentially, you don't even have to develop the wart. You can also develop a subclinical chronic infection that could lead to certain cancers of the cervix, vulva, vagina, penis, those the genital areas. And the two strains that are most problematic or cause the most cancers in this area are HPV 16 and HPV 18. It is presumed that they are responsible for 70% of the cervical cancer cases. Now these viruses, these are a little bit tough because they can't be grown in vitro yet. We cannot do a viral culture on these guys yet. Remember, they have a very specific host cell. So instead we can do cervical screenings using specialized smears. Of course, there's the Pap smear, okay, where you can look for the changes in the cell that point to a viral infection. But this won't give you the particular type of virus that's infected. Most cases of HPV will resolve on their own. It won't go anywhere. Within 2 years, you'll find that the patient doesn't have HPV growing in their wherever anymore. It's only a small number that will potentially produce this chronic situation that can lead to cervical cancer. Okay, so the Pap test is one of those ones that has been used in the past with pretty good, with moderate success. And then if you find, in other words, you've already found the cells that the virus has led to transformation. If you find abnormal cells with a Pap smear, then the patient has to have a colposcopy. It becomes much more invasive. So the theory was, let's move to more molecular testing where we could get more sensitive. We can see what type of HPV it was, so that we can determine the risk for this patient. So the patient might have HPV, but maybe it's not 16 or 18. A Pap smear won't tell us that. If it's not 16 or 18, maybe it'll resolve on its own. Okay, so that's kind of where we are. So there have been several molecular tests developed. The COBAS HPV test, which is just demonstrated here, where they create slides and then they do basically a little microarray on them. They've determined what the high-risk HPV strains are that they want to test for, including the 2 worst. So you've got 12 others, including the 2 worst, and you can see what they are here: 31, 33, 35, blah, blah, blah. And you'll screen all of them at the same time, okay? If your HPV 16 or 18 is positive, then a colposcopy is recommended, where the physician actually goes in and finds the area of growth and removes it. If any of the others are, cytology will come next, so that you'll do your Pap smear and you'll compare, or you probably did it at the same time, and see what those look like. And if they're all negative, they're negative. So come back to me in a few years. Now, the genes that are most significant in HPV that they actually act as oncogenes, are genes E6 and E7. These are the 2 that actually can change your cell cycle. And so in some cases, instead of testing for the strain of HPV, they test for these early genes and their production. So so I can test your cells and see if these HPV early genes are in there. So there's different theories about how you should test for this, because if E6 or in E7 are in your genome and they're in the right place, then there's a pretty high chance for you to develop cervical cancer. The BK and the JC viruses, these are polyoma viruses, and they're similar to each other, but clinically they're different from each other. Okay. Now, if I screened everybody in this room, most people would have evidence that they have had the BK virus at some point in their life. The infection itself, asymptomatic to mild primary infection, probably respiratory with a fever for that primary infection. And in either case, whether you have asymptomatic or mild, the virus will disseminate and it'll enter the kidneys, the urinary tract, and it'll persist for life in the kidney and the urinary tract. So you have a persistent chronic infection. And the thought is that about 80% of the population actually harbor the BK virus. So it develops what we call renal latency. Okay. Okay, so if 80% of the population, we're not all bad, what's the problem? Well, if the immunosuppression develops in that patient, and this is really where the problem comes. So if let's assume I had BK virus at some point in my life, and then later on I get cancer or I need a transplant, I'm immunocompromised. Well, if that occurs, particularly if the kidney or there's another organ involved, the this renal infection will progress to renal dysfunction, and you'll see a progressive loss of kidney function. You'll see it by progressive increase in serum creatinine because the kidney's not working, progressive though. And you'll get gradually worse and worse UA results. You'll see renal tubular cells, white blood cells in the UA. You're like, why is this patient getting so sick? And the reason is because of a reactivation of this BK virus. Now, this can be a problem with any transplant because we do suppress your immune system on purpose, of course, but unfortunately it can lead to what's called BK nephropathy, where the virus, this is post-transplant now. So you had the virus already, right? So in the first case, if you're immunocompromised, you could see progressive renal failure. In the case of a transplant, where then we make you immunocompromised to a certain extent for the rest of your life, the BK virus that was in your tissues already can enter the graft. And now the graft becomes the place where the virus can replicate. And that means you'll lose the graft. And the graft will be lost in about 80% of the cases if you get BK nephropathy. Okay. And the onset can occur literally within days of the transplant. So that would be absolutely awful. If we look at the bone marrow of patients who have BK associated issues, any of them, I'm sorry, not if we look at the bone marrow. We have also seen, if we look at bone marrow transplants, we can also see that that might also be associated with BK virus with hemorrhagic cystitis. So progressive decline of kidney function following doesn't seem related to anything else. Find out it's the BK virus reactivating. One of the things we look for are decoy cells. And if you look at these cells, they kind of look a little malignant because of the inconsistencies in their nuclear staining, right? You have little blebs on the outside in the cytoplasm. You have some inconsistent dark and pale spaces going on in the nucleus. You have some strange projections from the nucleus. These things, and the nucleus is of course also enlarged in there. So these are called decoy cells, and these are actually malignant cells from within the kidney that have shed into the urine. But you would only see these with staining, so these would not look like much of anything if you didn't stain it. Now the JC virus, also a very common virus. Most of us get it before we're adults, and it's thought that it's probably infected through contaminated water because it's found in high concentrations in urban sewage systems. So it's probably an infection of the tonsils or the GI tract. But the reality is we don't really know. We say that it's a generally asymptomatic or mild infection because we have no reason to look for it. You don't go to the doctor. It's such a mild infection, okay? But we do know that it does remain latent in the GI tract, and then also in the kidney. So you can develop a latency in both the GI and the kidney, or either one. And then it's still reproducing, you're still shedding it. And then what happens with reactivation is JCV will cross the blood-brain barrier and it'll infect the oligodendrocytes and the astrocytes of the brain. And you end up, there's a receptor on those cells that will accept JC. And once it's in those tissues, it'll produce what's called progressive multifocal leukoencephalopathy, or non-PML. Okay. Basically, it's an immunodeficiency that's going to allow this reactivation. It's not really reactivation in the true sense because it's not a true, it's not a dormant virus. The virus is still shedding into your urine. It's still a low level infection in both of these cases. But once your immune system pardon me, drops down, you end up with you end up with this situation where your immune system can't keep it in check and it starts to really run its course. Unfortunately, this will be fatal because of the destruction of the central nervous system, the material in there. There is some thought, recent studies are trying to link JC virus to colorectal cancer. It has been isolated from malignant colon tumors, but that's fairly new in research. HTLV1 and 2, these ones, human T lymphotrophic virus. These are retroviridase, single-stranded RNA viruses that do contain a reverse transcriptase. And they also target, much like HIV, its famous cousin, the CD4 positive T cells. It can infect other ones, but it does prefer CD4 positive cells. It will this particular virus will infect those cells, and then it will frequently become latent. At which point, meaning it will, reverse transcriptase into a DNA sequence, which it can be inserted into your own genome. It can remain latent, or it can become activated at any time. The things that activate it, not necessarily understood. Okay, and at that point, it can it'll complete its reproductive cycle. Okay. Pardon me. This is transmitted via the blood, sexually and vertically, and both of these viruses have 3 structural genes and 2 major regulatory genes. The structural genes are your GAG, your PUL, and your envelope genes. It's pretty standard for a retrovirus. And then there are 2 major regulatory genes called TAX and REV. these are the things against which we'll see antibody responses, okay? Now, the diseases that we see for these particular viruses, for HTLV1, this is actually endemic in not necessarily related areas of the world. It's endemic in southern Japan, the Caribbean islands, South and Central Africa, the Middle East, and parts of South America, where HTLV2 frequencies are highest in the Native American populations and in IV drug users in North America and in Europe. Okay, so HTLV2 is much less common than 1. HTLV1 also is associated with malignant and non-malignant disorders of the inflammatory or proliferative system, so T cells or macrophages, which it can also infect. One of the ones you've, I'm sure, heard of before is adult T cell leukemia or lymphoma, or ATL. This is the most notable risk of having this particular virus, particularly if the infection is acquired before you're fully adult. Okay, but the latency period is still extended, 20 years or more before you develop that. Okay. It's also associated with a progressive neurological disorder called HTLV-associated myelopathy tropical spastic paraparesis, which is sort of shrunk down to HAM, H-A-M, HTLV associated myelopathy TSP tropical spastic paraparesis. There you go. Okay. Just know the disease. Don't worry about what it is. But the risk of this also includes HTLV1. One of the reasons we get this is because HIV patients were developing this uveitis, which is associated with HTLV. They get this intraocular inflammation of the eyes. It's called HTLV uveitis. And it was causing a significant problem in HIV patients, so it was discovered there as well. Then you also have HTLV1 associated Zika syndrome, which is a version of it usually seen in kids, and basically these kids have dry eyes, dry mouth, and they have this dermatitis, which looks like a severe eczema. Now HTLV2, little bit, it's definitely thought to cause a less severe infection than HTLV1, but not in every case. It can cause a chronic encephalomyelopathy similar to HAM-TSP, and it can also cause a variety of generalized neurological disorders, as well as recurrent bladder and kidney infections, fungal infections, and pneumonia. But other than that, it does not have like a long list of diseases associated with it. There's no real gold standard for HTLV1 testing because these are more difficult tests to run. Usually these tests we usually send out because they're a little bit less commonly ordered. So we do have viral cultures that are possible, but they're really actually pretty complicated. So antigen testing has become much more the norm, okay? So we use ELISA testing to screen, similar to HIV, we use ELISA testing to screen the serum for antibodies against HTLV1 and HTLV2. So you're just looking if the antibodies are there. Then you do ELISA to determine what the antibodies are specific against. Okay, so it does require similar to, so you'll do a screen and then you'll do a Western blot to verify the screens or to specify the screens. Okay, and that's pretty much how we do it. The only caveat that CLSI requires is that if you end up with a reactive ELISA, so you note that the antibodies are there, but your Western blot, which we would normally consider a more sensitive test, but your Western blot is negative, then you have a problem. You have a discrepancy. And instead of just waiting 2 months and trying again, there's actually a more sensitive test called RIPA, which is radio immunoprecipitation assay, or you could do PCR. Either one of those are more sensitive, but you only need to do those if your ELISA is positive and your Western blot is negative. So finally, just as an overview, just to note here how we're going to diagnose these. If you know or if you suspect that there's a viral cause to your infection, you don't know if it's an RNA virus, if it's a DNA virus, you don't know what's going on with that. So what's our possibility? Well, a culture, viral culture, obviously that's one of the places where we can start. That's very generic, very non-sensitive. It usually will be able to detect most viruses that are growing. But it's slow, it's clunky, okay, and usually is not always. So another thing is to look with, well, if you have the virus, your immune system should know about it, so let's detect the antibodies. That's our next step. Again, it's easier, it's not clunky like a viral culture, it's not slow like a viral culture, but it's not always the most sensitive way, and especially if our patient is immunocompromised, okay? It's not necessarily the best way. So then we have to think about the molecular tests, targeting nucleic acids that are present, doing amplifications and sequencing that's present. Molecular tests is really the way to go in viral testing as we move forward in the laboratory.